Friends, welcome to Biology Made Easy. Today in this video, we will discuss pleiotropy. Pleiotropy is a Greek word derived from two different words. Pleion means many, and tropos means affecting. That means pleiotropy means a single gene that affects several characters. That is pleiotropy. So how it functions? Usually a single gene has or it regulates the several interrelated metabolic pathways which reflects in different phenotypes. Again, here another important thing. When different phenotypes are obtained due to action of a single gene, one should not think that all the phenotypes will have a major effect. In some cases, the phenotypic effects may be major. We can call it as a major effect. On some cases, the effect may be minor, and we can call it also secondary effect. That means when one gene affects several characters, several traits, all the phenotypic traits are not equal. Some may be greater, some may be less. Now, what are the examples of this pleiotropic gene? There are two examples from human beings. One is sickle cell anemia, the other is phenylketonuria. So, let us discuss. So, the first one that we will discuss, the first example, number one, is sickle cell anemia. This is one example of the pleiotropic gene. So in case of sickle cell anemia, what happens? We know that in our blood there is one cell that is RBC. The RBC in case of normal human beings, it is round and biconcave. That means it looks like this, round and biconcave. That means there is a depression in the middle. And the RBC contains one important protein that our pigment, you can tell this is hemoglobin. What is the main function of the hemoglobin? It usually carries oxygen. Now this hemoglobin, it contains two types of proteins. One is 85% is globin protein and 15% is heme protein. Now this globin protein, it is again up to different types. Usually this globin protein is a tetramer. It consists of two alpha chains and two beta chains. Now each chain of this alpha, it contains 141 amino acids. That means each one has 141, while each B chain has 146 amino acids. And the gene for synthesis of this beta chains, that is located in chromosome number 11. Chromosome 11. Now this is the case of this hemoglobin which is present. Now if this gene is mutated, what happens? This is the normal gene. If it will mutated, it will cause sickle cell anemia. How? Let us see. Suppose in one of the beta chains at the 6th position there is glutamic acid, because if you take this normal hemoglobin, in case of normal hemoglobin, at the 6th position, the DNA is GAG and it will be CTC. So this will give rise to mRNA and the mRNA will be GAG. And according to genetic code, the GAG codes for glutamic acid. So this is present at the 6th position. Then in this side there is 7th position, and this side there will be amino acid number 5, then 4, and so on up to 1. This is the normal one. Now if somehow this the thymine present in CTC, if it will be mutated and it will be converted to adenine, then what will happen? In that case, that will be converted to the sickle cell. Now this will be C, now it is converted to A, so CSC. So naturally this side will be G, T, G. That means you see, originally it was CTC, now it converted to CSC, so naturally the other strand is changed. Now when it will give rise to mRNA, now it will give rise to G and T, because in case of mRNA T is not there, so it will be GUG, and GUG will give rise to valine. The same at the same position, that is the 6th position. While all other amino acids remain same. That means in one of the beta chains, when at the 6th position, where glutamic acid is present in normal hemoglobin, if due to mutation it is converted to adenine, then the amino acid is changed to valine and it causes sickle cell anemia. So from this we can tell that the sickle cell anemia is a point mutation, because the mutation takes place at a point. It is a substitution mutation, because here the T is substituted by adenine. Then we can call it a transverse mutation, because in a transverse mutation, a purine is replaced by a pyrimidine, or pyrimidine replaced by purine. So here pyrimidine is replaced by purine. So it's a transverse mutation. And also it is an autosome linked recessive trait. That means when the character is present in the recessive, double recessive, then it will the person who will carry this double recessive will be will suffer from sickle cell anemia. Now when it is changed to this, what happens? Now let us see. If there is in one person there is a homozygous HbA and HbA, that means both the alleles are same, then the normal RBC. Suppose it will be heterozygous. Now it is heterozygous. So in case of heterozygous, one will be normal, the other allele is sickle cell. Now this person will not suffer from sickle cell anemia. Simply the person is a carrier of sickle cell anemia. Now again it will be homozygous. If it will be homozygous, that means both the alleles are mutated, double recessive. Now this person is the sufferer of sickle cell anemia, and this person hardly survives 16 to 18 years, and this person usually dies around 16 to 8 years, there is a death. So you see what is the difference between these things. Now if this person is normal, this person will suffer from sickle cell anemia and the lifespan decreases. But what about this heterozygous condition, when the person who is heterozygous for this condition is a carrier? Now in such cases what happens? What will be the condition of that carrier? That depends upon the availability of oxygen. If the oxygen concentration is normal, then there is no problem. But if in the atmosphere oxygen concentration lowers, as it happens when we go to some high altitudes or mountains, and there is a decrease in oxygen concentration. So when there is a decrease in oxygen concentration, when the oxygen concentration decreases, then usually what happens, the RBC becomes a sickle celled, like this. It becomes sickle celled, while the normal one is this one. When it becomes sickle celled, it becomes this, like this. So when it becomes sickle celled, usually it is destroyed in the capillaries and it causes some pain in the pain in the joints and in abdomen. So this happens only when there is a low concentration of oxygen, or in the atmosphere oxygen concentration decreases. Otherwise they are normal, just like the normal people. But there is another important thing. The sickle cell anemia persons, they can defend themselves from malaria. How? Usually the malaria parasite grows in our blood and it completes a life cycle. But those who are suffering from sickle cell anemia, in them the plasmodium, it cannot complete a life cycle. That is why it cannot cause malaria in the carriers. That means they are defending themselves from malaria. That is why in some parts of the world, like Africa, the Mediterranean parts, and in some parts of India, we'll find that in those areas which are pandemic to malaria, there are most of the people who are surviving, they are usually the heterozygous condition, they are the carriers of sickle cell anemia. And those who will be normal, they may suffer from malaria, but those who are carriers, they will not suffer from it. Because the plasmodium, it cannot multiply itself. So this is one example. Now let us take the second example, that is phenylketonuria. And here also you can ask one question: how can we know that whether a child is containing, whether it is carrier or not? How can we know? We can know it by blood or urine test. Or we can know it by blood testing. Because in the blood, if there will be two types of RBC, this is normal one, this is sickle cell, then it is carrier. If all will be sickle celled, then the sufferer. And if only this round biconjugate ones are there, then normal. So from that we can know from the childhood whether actually the person is normal or carrier or a sufferer of this sickle cell anemia. Now let us discuss the second example, that is phenylketonuria. The phenylketonuria is also an autosomal recessive disorder, and this gene is usually present in chromosome number 12. So the second example, we will take the second example, that is phenylketonuria. Phenylketonuria, in brief it is also known as PKU. So this is also an autosomal recessive disorder. It is found in chromosome 12, and when this gene is present in homozygous recessive condition, what happens? It lacks the synthesis of one enzyme, that is phenylalanine hydroxylase. That means when it is present in homozygous recessive condition, this enzyme cannot be synthesized. Now let us see what is the function of this enzyme. Usually in the body, the phenylalanine is converted to tyrosine inside the liver, and this conversion is carried out by this phenylalanine hydroxylase. Now, when this enzyme is not synthesized, that means in the body this phenylalanine is accumulated. So when it is accumulated, what will happen to the organism? Now, usually at birth, the person is normal. After some weeks, we'll find that there is a 30 to 50% increase in phenylalanine within weeks. Phenylalanine. As a result, what will happen? There is a mental retardation. That means there is no brain development. There is no brain development, we can tell. No brain development. And in 6 weeks, if the child will be not treated, in 6 weeks what will happen? 1/3 of the children, they cannot walk, while 2/3 of them, they cannot even talk. This is the seriousness of this phenylketonuria. And besides this, there is a decrease in pigmentation. There is a decrease in pigmentation of hair, skin, and also most of the time there is eczema. These are some of the characteristics of this disease. So these are the different symptoms of this disease, and usually when it is present in a recessive condition, these things happens. But if it is present in a heterozygous condition, then the person is normal. That means in heterozygous condition, the person is normal, but only when it will be homozygous condition, then all these things will happen. So, how can one know that whether this person is suffering or not? Again, by blood test or urine test. So, by testing the blood and urine, it can be known whether that this phenylalanine is there or not. Because the person who is suffering from this phenylketonuria, usually what happens in the urine as well as in the sweat, the phenylalanine is excreted. Phenylalanine is excreted. But in spite of this excretion, most of this phenylalanine is converted to phenylalanine pyruvic acid. Phenylalanine pyruvic acid and other metabolites. That means in spite of the excretion of this phenylalanine through urine and sweat, still there is a accumulation of this phenylalanine pyruvic acid and other metabolites, and they cause this retardation or underdeveloped brain. So these are the different types of symptoms which can be tested, and proper medication can be taken. So these are the two things, that is sickle cell anemia and phenylketonuria, that is coming under the examples of pleiotropy. Now let us discuss MCQs. So one MCQ, we can take the first one, sickle cell anemia. Sickle cell anemia is an example of is an example of first choice polygenic inheritance, second choice codominance, third choice pleiotropy, fourth choice multiple allelism. Now let us discuss the second question. The second question is a pleiotropic gene first choice has same phenotypic effect on different traits. Same phenotypic effect on different traits. Second choice has different phenotypic expression of the same trait. Then the third choice influences phenotypic expression in different traits. And the fourth choice is inhibits phenotypic expression of other traits. So these are the two MCQs, and such type of questions are usually asked in all types of national tests. So thank you very much for watching the video. God bless you. Have a good day.